Hi, I'm James Maclaine, and I'm one of two fish curators at the Natural History Museum in London. As well as looking after our existing fish collection, which includes hundreds of thousands of specimens, some dating back to the 1760s, I'm also occasionally lucky enough to get to go out and make new collections as well. In 2019, three of us, myself, Jonathan Abbott and Kirsty Lloyd, were very happy to be invited on an expedition to the South Atlantic as part of the UK government's Bluebelt programme, which was being organised by the British Antarctic Survey and CEFAS. I would be processing fishes, John would be doing cephalopods, and Kirsty would be taking tissue samples. The Bluebelt programme aims to survey the marine environment around UK overseas territories, an area of over 4 million square kilometres, and to improve our scientific understanding so that areas of protection can be set up, particularly with regard to managing existing fisheries to make them as sustainable as possible. This particular expedition was going to look at Tristan da Cunha and St Helena with a broad range of objectives for each territory. For Tristan, the survey looked at biodiversity and the habitats associated with the surrounding seamounts, the main area for demersal fishing. We were very happy to hear last year that a marine protected zone of nearly 700,000 square kilometres was set up around the area as a result of our trip. The St Helena leg focused on pelagic ecosystems with a view to improving our understanding of the food webs that underpin tuna fisheries around the island and Cardinose Seamount to the north. This shot from Google Earth will give you an idea of just how remote the islands are. 1. The Falkland Islands, where the voyage began. 2. Tristan da Cunha, the world's most remote inhabited island apparently, home to around 250 people. And then, 1,500 miles away is St Helena, number 3. The whole trip was well over 5,000 nautical miles and took around 5 weeks. Here's our ship, the Discovery, a very comfortable vessel fully equipped with lab space, scientific equipment, a cold room for specimen preparation, and even a cinema. This is the business end. This is where we lower the gear, a massive rectangular mouth trawl made up of 2 nets, each with a 25 metre square mouth that can open and close on demand. What we mostly did was lower everything to 1,000 metres and then trawl between there and 700 metres for about 40 minutes, and then open the second net and do another trawl up to 400 metres, so each event would yield 2 samples. It looked a bit like this. This is a very small one made as a test in the open sea, and it got a bit bigger when we trawled around the more productive seamounts. This is a very common kind of mid-water fish called a lanternfish. We got loads of these in our trawls. They're covered in tiny light, which you can see still glowing very faintly blue in this specimen. This genus Diaphis is distinctive for having 2 large headlights just in front of its eyes. A much more predatory fish, Sloan's viperfish, with fangs so big that they cannot fit into its mouth but slide up the front of its face instead. It also has lights along its body, but also a modified fin ray, which it uses as a lure to attract its prey. This is a sample from near St Helena where the waters are more productive, so we got much more diverse samples. There are at least 40 species of fish here. More predators, a fangtooth and a selection of juvenile deep-sea anglerfish which also use lures for catching their prey. This is one of my personal favourites, an incredibly rare anglerfish called a Toothless Sea Devil. Only a handful of these have ever been caught, and it is a complete mystery as to why they've lost their teeth. Nobody knew what they ate either, so it was very exciting to catch one with a full stomach. I'm currently investigating this using CT scanning. It's definitely consumed part of an eel, but there are other remains in there too which I'm trying to identify. These were, I think, the biggest and weirdest fish that we got, inflated whiptails. They're also very rare, and these 2 specimens now double the total number in the NHM collection. So all these specimens, nearly 3,500 lots in total, and their 2,000 tissue samples were all brought back to the NHM where we curated and databased them, and then made them available for any interesting science projects, which is where Alex comes in.
Thanks, James. Hi, I'm Alex and I'm a PhD student at the Natural History Museum and at Royal Holloway, University of London. I usually research plastic pollution in the River Thames in the UK, so this was a very exciting change to my routine. James provided me with a selection of species including the Sloan's viperfish and the inflated whiptail you've just seen. Overall, 32 fish were examined from 15 species. These were mostly mesopelagic species, but we also sampled 2 shallow water species for comparison. To recover the plastic, I firstly removed the digestive tract. In the top right image, you can see this black structure, which is the stomach of Opostomias. Any prey items in the digestive tract were removed, identified, and analysed separately. This allows us to estimate contamination in the prey species as well. The digestive tract and prey items were digested overnight in a 10% solution of potassium hydroxide at 50 degrees C. This removes most of the organic material, making it easier to find the plastic. In the bottom image here, you can see that the solution is clear, with only a small amount of material which has settled to the bottom of the tube. This solution is then filtered, and that filter searched under a microscope. More mesopelagic fishes contained plastic in the digestive tract than shallow water species. We only had a small sample of shallow water species, just a couple of specimens collected on the discovery expedition. So whilst we didn't find any evidence of contamination, it is still likely that shallow water species are also affected. Most of the plastic recovered was fibres, and you can see a couple of examples of those here. 2/3 of the mesopelagic specimens were contaminated with microplastic. For comparison, an average of 28% of Thames fish have plastic in the digestive tract, and worldwide commonly about 1/3 of fish are contaminated. Again, we only have a small sample size in this study, but from these specimens it is clear that plastic penetrates to the deep ocean. We couldn't compare plastic ingestion between species due to our limited sample. Instead, we grouped fish based on their feeding behaviour. Predatory species ingested significantly more plastic than planktivorous fish and benthic feeders. This could be due to the trophic transfer of plastic from prey to predators. This is why we removed the prey items before digestion. Here you can see 2 fangtooth collected by James. The individual in the top image clearly has a full stomach. Indeed, when I dissected this individual, we found many intact prey items, likely the result of net feeding. Most notably, there is a cock-eyed squid, a sea devil, and a bollitinid octopus. Examinations of the prey revealed that both the squid and sea devil had ingested microplastics. If these prey items had been digested, the plastic would have been transferred to the fangtooth. The plastic recovered in the mesopelagic fishes were polyester and cellulosic fibres. The cellulosic fibres include cotton and a semi-synthetic material known as viscose. Viscose is a modified form of cellulose, often used in sanitary pads and wipes, as well as in textiles as synthetic silk. Many of these fibres likely originate from washing our clothes. An average wash can release 700,000 fibres. These exit the machine in wastewater and are transported to plants where they are filtered out even further. Many plants can successfully filter up to 99% of plastics in the wastewater. However, that 1% worldwide quickly adds up. Another source of plastic that these fibres could originate from is sewage-related debris, things like sanitary pads and wet wipes which wash down the toilet. These can then fragment in the environment. We can stop this plastic entering the environment and being ingested by fish by implementing more filters in washing machines or in wastewater treatment plants, or perhaps designing clothes that shed less fibres. Sewage-related debris could be stopped by people not flushing sanitary pads or wipes. Perhaps this means there needs to be better labelling on those products, or education as to the impacts of what happens when they are flushed. It's hard to isolate the exact source of plastics in the ocean. Models have demonstrated that microplastics released from South Africa can be transported to the South Atlantic, so plastic in the deep ocean could have come from many sources. That's all from us. We'd like to take this opportunity to acknowledge all the support we had in this project, and we'd like to thank everyone for watching.